Well, okay, my friends, yesterday I did a video about Johns Hopkins and Yale getting together trying to find new treatments for basically for cancer tumors. And they're leaders in the cancer research, and they're teaming up together here. This just came out less than a year ago. And they're talking about some serious problems with these survival rates, where 5-year survival rate for pancreatic cancer is less than 11%, and melanoma is not good either. Now, I did go to Johns Hopkins, as I mentioned yesterday, for genomics, so I have some understanding of this, and I am very, very deep into the chemistry of the biological components, specifically enzymes. And enzymes only happen because of bacteria. So we're going to go right through the chain of events for the last 10 years or so in the study of this enzyme response to invasion, and to also breaking down the molecules in your body to clean out the trash, to prepare the food. I mean, they do everything. The enzymes, they're like everything, and they are so elegant. It's absolutely, look at this. I'm going to just show you this, and then we're going to get started. This is what bacteria do. They create enzymes. And these enzymes are just unbelievable. Look at this. This is what an enzyme looks like when it's finally constructed and done. And they squirt out of the back of bacteria just like this. And when they get to a certain size, they just go. And then that's exactly what they will be like for the rest of their life. And they only last a certain amount of time. They don't stay forever. They go out and they do click chemistry. They're exuded by bacteria. They shoot them out in response to invasion. And they have just pretty much determined this in a new video. Watch this. All right, you should watch this whole video. Very interesting. It's called Your Textbooks Are Wrong. This is what cells actually look like. Now, this goes back a couple of years, 3 years ago. It says, you probably remember being taught about the cell in your high school classes, but it's not anything like they taught you. Now, this is Seeker, and this guy's name is Gokal, and he's from, I believe it's UC Berkeley. And here's what they found. This is called Your Textbooks Are Wrong. This is what cells actually look like. And again, it's from Seeker. Listen to this.
I still remember the moment. It's something I will never forget. The hair on my hands just stood up. It's a microscopic universe within each cell.
This is an unprecedented view of the cellular world, where we can actually see immune cells scooping up sugars in the ear of a zebrafish in real time.
Focusing only on the crawling immune cells, we've noticed two classes of them. One was not hungry at all, but it was very active in terms of trying to figure out, what the environment is. There's another one that was kind of slouching around with a lot of food in its belly. We can actually conceptualize and visualize and analyze the contents of each of these cellular compartments in this crawling immune cell as it's kind of, scooping up its environment. I mean, that is a level of details no one's seen before.
We're living in a new era of cell biology, where microscopy advancements are giving biologists the opportunity to reveal the hidden patterns of cells. What we expect to learn from here on out will transform our understanding of human health and rewrite the textbooks on the fundamental unit of life.
I agree. Okay, so my outstanding friend Gokal is saying that they will rewrite the textbooks on the fundamental unit of life, which is basically the cell, and every cell and every organ is protected by a membrane. That is what we have to rewrite, is how that membrane reacts. Now, we're going to go back and look at what he had to say, and I'm going to focus in on each one of the little spots that he talks about, the different types of, well, let's go back and look. All right, he says it's a microscopic universe inside each cell. All of these are different cells in the body. It's just staggering the amount.
Microscopic universe.
It's a microscopic universe.
This isn't...
Okay, now, here we are inside a cell. All right, now, they're going to actually see the bacteria in here with a certain type of light, which lights it up.
Unprecedented view of the cellular world.
Now watch, see? All right. Here we are inside an immune cell, or a leukocyte, which is your white blood cell. So this is, they can see inside the immune cell scooping up sugars. Now they say sugars, well that's fine, that could very well be. But I believe this is a bacteria. And it is running around here grabbing up all its little enzymes or whatever it's grabbing up.
Where we can actually see immune cells scooping up sugars in the ear of a zebrafish in real time.
And did you see that thing squirting out? That went right out through what they call a ion channel, which allows it to get out of the cell membrane. Let me show you what cell membranes look like. Hold on one second. All right, these are the kind of membranes where you saw that little thing squirting around in there and zip, it went squirting out. Probably through a protein channel, because that looked like a protein, which is an enzyme, or at least maybe it was a bacteria carrying an enzyme, I don't know. But the bacteria have the flagella. They can just go squirting all around inside the cell and then grab up the protein. Something grabbed up the enzymes and zip, it went shooting out to the other side, out into that matrix to go deliver that to kill something. That's what I'm taking away from the latest research, is the enzyme is literally 100% of the chemistry in your body. Here's the deal. Here's the deal with your cells and your bodily tissue. All the toxins, antibiotics, pollutants, and all that stuff, they kill bacteria. Bacteria create the enzymes. The enzymes were those elegant little molecules. Here's the enzymes right here. Hold on. That's what the enzyme is. That comes out of a bacteria. And here's what a bacteria is. You see that? And here's what happens. That bacteria is squirting around through your body, and somebody gives it a command, which is that other bacteria that says, hey, I'm just out sniffing. We're going to go back and look at that. And it says, go out and squirt out that chemical that you usually squirt out. And it says, okay, no problem. We got plenty of that. Squirts all this little chemical out. And then when it comes back here, it goes, and then all of a sudden it's a protein. Very, very elegant molecule. And then it goes floating through your system, or it's probably right at where it's supposed to be, because these things have flagellas. They can go a bazillion miles an hour through your body. The flagellas on these things can spin 100,000 RPMs. And it's called a fluid-filled highway. This interstitium, which they really just now are starting to understand how dynamic that little space is. I suppose I should show you that too. This gets very involved. But the basis of the whole thing is we have bacteria that have to be living in you, and there are trillions of them, and there's thousands and thousands of species. And every one of them creates a chain of, like, this is what they do. That's their job, is to create proteins. And the exact sequence, the DNA is the program. It says, hey, when you're going to squirt out that stuff, somebody tells you to squirt it out. Here's what I want you to do. Put 2 green ones and a red one and a blue one and an orange one, and they just go squirting out. And then it says, okay, that's it, cut it off, click. And then it all binds together into the cell, into different ways in the DNA, RNA, and so forth. That part we're not going to discuss. We're going to discuss why these are alerted, how they know what to do, what they produce, and how it interacts with everything else, because it is a very, very aggressive click chemical, and it is unbelievably sophisticated, as you can see right there. This is what happens. All of that came squirting out in one big long strain, and then went, and now it's got a plus and 2 minuses and 6 minus pluses, and that thing goes floating through your body, snap. And when it hits other chemistry like this big attack molecule, it goes bam, and everybody that was attached to that attack molecule is infected basically by that magnetic impulse, and it goes boom, and all these things break and they become what they call denatured. They no longer do their natural thing and everything dies. That's what's supposed to happen when an enzyme comes and attacks. Okay, you've just seen that they really didn't know what cells look like. This is live science, and this is about the new meet your interstitial, the newfound organ, which is this layer that I'm talking about. And this layer right here, the dark layer, I believe is the fluid-filled highway that all of the bacteria and enzymes and so forth transfer through your body through that layer of membrane, because that's all it is a membrane. This is like the outside layer of your skin, or any outside layer or any outside membrane of an organ that's in the body. They're all covered with a membrane. So this is the membrane. Could be skin, could be mucosa, could be some kind of fabric like on the lung, pleura. Between that fabric and your body, which is your vital tissues under here, is a barrier. It's a literal barrier. It's a membrane. It says, I'm only letting you guys come through that I want come through, and the rest of you stay the hell out. And if you try to get through, I got guys here that are going to kill you. Now, in addition to that, there's secretions out of this zone as well that attack food and break it down, make all the products in your body to keep you healthy. All right, and all of that stuff happens as a result of bacteria. All these things are saturated with bacteria that create enzymes. And what it is basically, as I showed you before, I'm sure I showed you this, the bacteria create the enzymes do the chemistry. They go out and break things down hundreds of thousands of times in a second, versus they would never happen in real life if it wasn't for these enzymes. And this is a kind of elegant little enzyme. What are enzymes? Are enzymes proteins? Yes, they are. Proteins come from bacteria. Enzymes come from bacteria. And here's an invasion of some kind of virus or whatever it is, and being attacked by, it looks like to me, like maybe even white blood cells. I don't know. Some of this stuff I don't know about. I get people sending me things from researchers all the time, don't want to be associated to anything outside of what they've been told to say. So I get these things and I don't even know exactly where they came from. But it looks to me like these are fighting against these. Something's going on in that interface. And again, like this almost looks exactly like that. You see that? They're trying to fight into that. To me, looks like the reality. This is obviously looks like a rendition of a drawing of it. But the other one looks real. Well, I mean, it is. I can say it. If somebody faked that, I doubt it. What it actually represents, I believe, is an invasion of some kind of tissue. Now, what is this down here? It looks like it's some kind of fibrin fabric just within your body. These are extremely magnified shots. And oh, by the way, everybody's been asking me about my big book, which is very, very good. I mean, I have to tell you, that thing is just incredible. Anyway, I'm going to give you the ISBN number for it in here somewhere, so stick around if you want to get that number. Now, every single cell in your body, including blood cells, have these barriers, and they only let things in and out that they want to come in and out. That's how it works. They're protein channels. So a protein is the enzyme. The enzyme goes out and does its work, just like we saw from Gokul. Now let's go back to Gokul's video. I'm going to discuss exactly what he shows, which is new information. I'm going to tell you right now, very, very, very few healthcare providers understand this at this point. So this is why this is research. This is not to say do this or do that. Let's all think about what we may have missed. So don't forget, this is what we're trying to prevent, is invasion through the membranes. How do we prevent that invasion? That's what we're trying to figure out. Okay, now this is the war against superbugs, and we all know about them. We just had a very, very bad round of it, and it's still going on, I guess. Some people adapt pretty quickly against these things, and they get healthy again. Some people don't get healthy for a very long time. They call it long COVID. So we're going to look into that too. But this superbug situation could have a lot of underlying factors. All right, just to make sure you understand, this is very, very new research. They didn't even realize this layer existed here before, because every time they saw it, it was in slides and it was all flattened out. They thought it was a flat layer. See, you wouldn't expect doctors to discover a new organ part in this day and age, but now researchers say that's exactly what happened. They found a network of fluid-filled spaces in tissues that hadn't been seen before. Those fluid-filled spaces are the fluid-filled highway that sends all of that, well, here it is. They found a network of fluid-filled spaces in a tissue never been seen before. The fluid-filled spaces were discovered in connective tissues all over the body, including below the skin, the surface, the lining of the digestive tract, the lungs, urinary system, surrounding every single muscle. They are membranes. And the membrane, according to a new study detailing the findings, they detailed this new layer that they had never seen before. And see, previously researchers had thought these tissues were a dense wall of collagen, a strong structural protein. Protein, again, made by enzymes, found in connective tissues. The new finding reveals that rather than a wall, this tissue is more like an open, fluid-filled highway, said the study's author, Dr. Neil Tice, a professor of pathology, New York University. Now, I know people that know him, and I've been looking at this in my Mudfossils, which I've been looking at for quite some time, and sort of trying to understand what this is about. And it's a good thing they discovered, because I believe this is the immune system, basically, right there. And if you don't have that stuff in that fluid-filled highway to go deliver the stuff, remember they showed Gokal showing these pictures of these white blood cells? I believe they're very, very similar to what you're seeing right here. Okay, we're back at Gokal, and this is one of these bacterias, it appears to me, that's scrooshing around here, grabbing up all of these little bacterias or enzymes or whatever they are, but it looks to me like it must be that single-celled bacteria. And it's running around grabbing up enzymes. I'm going to probably have to go with that, because then it goes out through these protein channels, one of these channels up here. Now, let's watch it. Remember, it's running all around here. Well, let me go back just a skosh here so that... All right, so here it is. It's just an empty space in here. It gets a command. Go in there and scoop up all of these little particles that are enzymes.
Representative view of the cellular world, where...
All right, now watch. This is the thing here, which I would believe is the bacteria. And it's grabbing up all of these things, and then it's going to shoot out of there like a rocket ship, because it has, I believe, a flagellum on there. When I watch,
where we can actually see,
you see the little
cells scooping.
There was one shot there, you could see the tail.
Up sugars in the ear of a zebrafish in real time.
Zip.
Focusing only on the crawling immune cells, we've noticed two classes of them.
All right, here he's saying this. We've noticed two classes of them. The first one is a sniffer.
One was not hungry at all, but it was very active in terms of trying to figure out, what the environment is.
Look at this. How bizarre is this? This is exactly what he's talking about. It's going around and saying, anybody match this string of beads? And it says, ooh, I see some really bad guys over there, and they match this. I went by them and I could see that, ooh, 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 and they attached there, and they attached there, and they attached there. I know these are bad guys. This guy goes back to the other guy and he says, make a bunch of the chemistry to go kill this particular guy. And that's what an enzyme is.
There's another one that was kind of slouching around with a lot of food in its belly.
That's the one. Watch, it's going to shoot out of here. And I believe it's got a flagella on there, and so I'm pretty sure that's a bacteria.
We can actually conceptualize and visualize and analyze the contents of each of these cellular compartments in this crawling immune cell as it's kind of, scooping up its environment. I mean, that is a level of details no one's seen before.
We're living in
We are living in a new age. There's no question about that. All right, I'm going to home in on a couple things, but just so that we can clarify for certain you understand. This is the bilipid layer. It's a 2-layer membrane. Everything in your body has that layer, and only allow things in that it wants to allow in and keeps the rest of the stuff out. It has to have that layer. It has to be in good condition. These little cellular junctions, you see all these little dots? They have to be held together in what's called tight junctions. They got to move around, but you don't want them opening up so something can come through. It's not supposed to come through unless the channel allows it. So if you have loose junctions instead of tight junctions, you're in trouble. Now, there's a bunch of different reasons that can happen, but primarily it's chemistry. If you don't have the right chemistry to create the right glue, literally, it's glue that keeps these things together in these tight junctions. So there's a lot to think about, but anyway, that's the membrane. And once this thing here attaches and it can beat and eat through the membrane, you can see this is a very good, I almost could believe that was real, but I think it's a fake. This, I say, is real. Virtually the same thing. So anyway, and this is the complexity of the molecule that we're dealing with, and it all shot out from a bacteria. Thousands and thousands of different bacteria in your body. And they create trillions and trillions and trillions of particles of enzymes, probably per hour. The process of life is so complicated, and nobody has a clue. This is, again, this is the bacteria. These are bacteria. This is an interaction between, I believe, white blood cells and invading virus or whatever. This is the interstitium that they talked about. This is a fluid-filled highway they never realized was here. And all these little black balls are anchors. So this can float around and go this way and that way and come back. That was not understood until recently. And this is from the most recent research on interstitium. This is, looking at it from kind of a ways off. This is a layer of skin membrane, actually, and then the skin up above. This is the fluid-filled highway. Now, this is closer up than that. So now we're into a closer, tighter shot over here. And this, again, is with the fluid-filled highway. All this area they run through. And it's all got to be able to still be able to be flexible, but still be a highway. And then you get down to where you can see how it can be flexible, but you have all of this fluid spaces between. These bacteria are very small. They can squirt. They're single cells. But they have some capability. These little bacteria, it's unbelievable how elegant the chemistry they make is. It's just incredible. Absolutely stunning. That little bugger, these things, and there's all kinds of different looking bacteria. Some of them have fuzz all over them. Some of them have one propeller in the back. Some of them have 10, all kinds of stuff. Some of them have spikes on them. Some of them, they're just a vast variety. Let's go with that. So you have to have a vast variety of bacteria in you that won't let the bad guys get in. And you have to maintain them, and your body has to grow them and keep them protected and keep them in these membranes, these fluid-filled highways to protect you. Now, let's look at what enzymes actually do, and what have they actually found out about these enzymes and about these membranes and about these tumors. Let's take a look at the research. All right. Again, Yale, Johns Hopkins, they're doing a lot on this. So are a lot of other people. But I went way back to build a complete profile of what has been done. Because some things might be missed. All right, this is the most important thing to take away from this. Chemistry, trace, decomposition, chemistry. What does that mean? Protein degradation. Those are these enzymes. Bacterial enzymes are responsible for the degradation of proteins. They break up other proteins. Bacterial enzymes are proteins. But they also break up other proteins into their component amino acids. That's their job. They go out and break up other proteins. Enzymes are proteins. The process is known as proteolysis. And it occurs at different rates for different types of proteins in your body. Now, that is and everywhere you look, they're talking about bacterial enzymes. They modify other substances. Anti-infective therapy, bacterial enzymes. This goes back quite a ways. They've been looking at this. Membrane damaging phospholipases and all kinds of things. Now, I've gone back and looked at a lot of the studies, and they all have one thing in common, is that they're all related to bacteria. They're all related to bacteria. The only thing that causes chemistry in you, virtually the only thing it does, is an enzyme, which is one of these elegant proteins. It goes, and then it goes out and it does chemistry based on its magnetic signature. This has been missed, I believe. And also there's another thing that's been missed, is the junk DNA, which is really not junk. It's programs that are stored of everything in your life in these little, what they call CPG islands. You know I bring into a lot of things into this, but we have to understand it in totality, or it's not understandable. Where are all of these commands stored? How do you react to different environments? Normally is related to what happened to you in the past. Something is imprinted upon you in these memory banks, which are your junk DNA, because everybody's is different. So they say, oh, all this stuff is structural DNA, and from here to here, we understand that, but all the other 95% means nothing, because everybody's is different. Well, that's not the case. The case is that yours is different than mine because you had some other event in your life, or you were sick, or you were attacked, or you were scared, or you were hungry, or you were loved or unloved, or who knows what. But that's how it happens. And identical twins at birth, their junk DNA is identical because everything about them is identical. And then almost instantly, they start to change the junk DNA. A little bit at a time. But if they're completely separated at birth, they just completely change because they're exposed to different foods, different interactions with people and animals and who knows what. So there's a lot to take into consideration here, and a lot of that is also stored in these programs. So we can't just ignore the programs and look at the enzymes. We have to look at the whole thing as a program needs an enzyme to carry out its command. The program is your DNA. And junk DNA is nothing more than normal DNA. Normal DNA tells you where your thumb goes, where your eyeball does, and where these muscles attach. Very, very enormous amount of instruction code. And then they say, oh, 95% of or whatever it is, a huge amount, is is just junk. It doesn't do anything because it doesn't manufacture something. Well, it is your memories. It's everything else about you. It's your immune system. It's how your bacteria can go and ask for instructions how to deal with invaders and how to, to me, that's what it looks like to me. So these are all things we have to take into account as we move forward, trying to understand why these tumors invade and why we can't stop them and why some people can stop them. And to me, it boils down to the bacteria creating amino acids and the enzymes that make your life function. Okay, my friends, as you know, basically everything I'm talking about right now is enzymes, which are produced by bacteria. Now, this is my medical book here, which everybody seems to be quite taken by, so I am going to give you the ISBN number, and if you'd like, you can order one. It was very inexpensive, as I recall. All right, the organs in your body, every one of them creates enzymes, basically. All its secreted bile and enzymes, pancreas, liver. Look at this. I showed this the other day, but I'm just going to run through it again, because this is where enzymes are made. Every one of these organs makes a certain set of enzymes, and they are quite elegant. Every organ in your body is so complicated, the average person just can't even, it bounces right off them. It's just too complicated. Look at this. Hold on. Check this out. Look at this. These are all the different types of digestive areas in the digestive system. Can you see this? I believe so. Yeah, and if you look through them, it's, every one of them has a barrier, this barrier. You see it? Every one of them has that barrier that it is the protective barrier. And they call it the epithelium. Well, the epithelium is now called the interstitial fluid highway. All right, so the epithelium was at one time just considered a flat plate. Well, it isn't. This book shows them exactly the way they are. I mean, this is exactly what they look like in the body. Very, very cool. And an infant has really no digestive system. So you have to be very careful about infecting them with something because they have no real way to fight back. And then you got into how complicated these things are. Gallstones and gallbladders, enzymes, pancreatic. These are different types of gastric ulcers and things. Tunica that surrounds the stomach and stomach lining. I show a lot of this stuff in my fossil studies. Anyway, it's a very, very complicated system of biology. Okay, this goes back 7 years ago, and this is about fecal microbial transplantation for C. difficile, which now they are allowing, but only if you can't take the drugs. You have to fail with the drugs first before they'll allow this, which is kind of disturbing. But anyway, here's what's going on now. This is treating C. diff with fecal microbial transplantation, which is nothing more than replacing your digestive system enzymes with the bacteria that create the enzymes. So here, I'm going to just let it play and watch what they have. Again, this is Fair Use Act. I'm not trying to steal anything, but this is what they're saying about this 7 years ago at Johns Hopkins.
Transplantation. A healthy digestive system is home to thousands of bacteria. The intestinal tract includes the stomach, small intestine, and large intestine, which is also known as the colon. In most cases, these microbes are harmless or even beneficial to a healthy individual. However, conditions that require treatment with antibiotics can kill off many of the good bacteria in the colon, allowing the bad bacteria, called Clostridium difficile, to take over. Toxins released by Clostridium difficile can damage the lining of the colon, a condition known as Clostridium difficile colitis. Clostridium difficile infection can lead to life-threatening complications, such as toxic megacolon, where the colon becomes dangerously enlarged. Clostridium difficile is normally treated with a course of antibiotics, but recurrent infection occurs in between 20% to 60% of patients after an initial episode. The standard treatment for a recurrent infection is antibiotics, but in cases where this fails, there is a new alternative treatment. An emerging solution for patients with recurrent infection is fecal microbial transplantation, or FMT. Also known as stool transplantation, FMT restores good bacteria in the colon by using the good bacteria from a donor. Stool samples are obtained from healthy donors and are screened to ensure there are no infectious diseases. There are different ways to introduce the good bacteria, but one common way is with a colonoscopy. First, the stool is prepared by blending it with normal saline, which is a saltwater solution. Using a colonoscope, this mixture can be applied to the entire length of the colon. Studies have shown that over 90% of patients treated with fecal microbial transplantation are cured with no significant side effects reported to date. Please speak to your doctor about fecal microbial transplantation if you are considering this treatment.
Okay, you see what they said? Over 90% cure rate with no adverse effects. Now, this was 7 years ago, and prior to that, they were looking at probiotics, which are the enzymes, basically. And sometimes they're viable living bacteria, but I believe most of the time they were giving you supplements that were the things that hopefully would have been produced by these little critters. But when your little critters are dead, you're not going to have that chemistry. You don't have that chemistry, you're going to be sick. And these people with the C. diff were being invaded, and they had nobody to fight back because they had some antibiotics that killed the bacteria that was supposed to be living in that layer that I showed you before, that interstitial layer. So let's see what other research has been done since this, and even at this same way back then. I went back to, I think, 2003, and there was some pretty interesting research done that seemed to be so disparate. It just seemed to be so, one over here, one over there, and one over here. Nobody ever got together. So I'm trying to put it in a big ball. Let's see what I got. Now, again, this is Johns Hopkins, and this was, again, 7 years ago, this video about this FMT, and it said, at this time, which is 7 years ago, FMT is not clinically performed for indications other than C. diff. More research studies are still needed to determine if FMT should be performed for other clinical indications. They think at that point they realized there was a benefit in all kinds of areas, but it's only for recurrent C. diff, which means that you have to fail the drug treatment in a number of times before they'll allow you to have. And it's still that way now. You can't just go get a fecal matter transplant for anything, unless your doctor says, oh yeah, he failed the treatment enough times that we'll let him get healthy. Which, because right now it's a 90% cure rate. As you saw, I mean, back then I think it was 90%. I think they mentioned it in that video. So why would they wait until you're ready to die before you can have it? Doesn't make sense to me. All right, now we know about the decomposition chemistry in these proteins, these bacterial enzymes, and they break things up so your lymph nodes can discharge them through your excretory system and get rid of them out of your body. Otherwise, they build up and break through the walls just like this. And then you have nothing here to fight back against them. You have to have these things saturated with those bacteria. That's why one person stays healthy, another person gets sick. That's what pre-existing conditions are all about. Your membranes here have weak junctions. All right, so don't forget, right here is a very good example of a lot of things that can go wrong. If you don't have the right proteins to go out to do the job, you're going to be sick. If you don't have the right chemistry to keep these cellular walls tight and junctions tight together, you're going to get invaded through this, and it will break through, and it'll continue to break through and break through until it gets through. Now, there's all kinds of chemistry going on in between the layers, and these layers are not just one little layer. There's a lot of layers there. They really never realized this, and this interstitium is now starting to be understood. All right, just let's review it one time quickly so you understand there's a whole lot of things going on. Way up even in your throat, your salivary glands and so forth, that's enzymes. Those are enzymes. They break down the food. Well, to some degree. Your stomach has all the acids. It really does the job. And then it comes down here into your small intestines, and that has a whole way of dealing with the stuff that comes through there. I'm not exactly sure what it takes out of them, but they call it jejunium and all these different little names, duodenum and so forth. But anyway, then it gets down to your large intestine, and then it ends up getting down to your anus, and where you poop everything out. So, and then you have like an appendix. There's something, the appendix, I believe, is your backup bacteria. So if everything out here gets killed, whatever's tucked away in the appendix is supposed to get you back up and running again. That's, I believe, what the whole idea. And appendicitis means something went wrong with that bacteria. Now that might even be a good spot to introduce something. I don't know. There's a lot to be understood now about this whole system of what enzymes are created where, and what do those enzymes affect, and what bacteria do you need to create those enzymes, because you need bacteria to create the enzymes. You don't have the bacteria, I don't care what you do, you're not going to have the enzymes. Bacterial enzymes, that's it, my friend. Look at this. So I'm really going through the whole range of different afflictions, and what have they found out? Look at this. Uveitis is inflammation inside your eye. It's when your immune system is fighting an infection. Well, guess what? The gut and the eye. This is the American Academy of Orthodontology. In today's common usage, microbiome refers to the community of bacteria, viruses, fungi, and yeast, plus all their genetic material that live in or on the body. Microbiota refers to one type of an individual microbe. All right? Just how complex is the gut microbiome? A estimated 1,000 species of bacteria alone live commensally in the human digestive system. Well, there's probably a lot more than that, with 10 times the number of genes in the human genome. 10 times the number of genes. They are creating bacteria, enzymes that are just absolutely incredible. Only a few of these microbes are well understood, and virtually none of them. It's as if in a vast orchestra, only a handful of instruments can be heard. But these early notes may lead to new therapeutic paradigms where the gut microbe plays a role in treating eye disease. This is the gut and the eye. They realize microbiomes. Look at, they're talking about the human gut, 70% of the immune system. They are starting to fully understand this, but it's a very glacial pace that they're accepting it. This is dermatology, World Journal of Dermatology, fecal matter microbiota transplant and dermatological disorders, a retrospective study assessing the gut microbiome's role in skin disease. And it comes down here to say that there is indication that fecal matter transplants has the potential to alter the course of chronic skin disease, alter the course. But few studies have investigated the phenomena beyond case reports. That's where I'm coming and trying to put things together. So they have a few case reports, but that's it, so they just sort of dismiss it. Research with larger sample sizes is needed to provide a more thorough assessment of possible associations and to establish a broader foundation upon which to base hypotheses. And they just haven't done that. All right, what do we have here? This is, Crohn's disease. This is in your gut, where it's eaten through your guts. Systematic literature search was performed through to August 2020. Studies were included if they reported FMT administration in patients with Crohn's disease and reported on clinical outcomes. And yes, it was very, very, here it is right here. No serious adverse events were reported. Some of them were like 75 to 100% effective in Crohn's disease, which is like that, very similar to C. diff. And what do we have here? This is colitis, same sort of stuff, eaten through your guts. Fecal matter transplant, followed up by anti-inflammatory diet alone is effective in inducing and maintaining remission over 1 year in mild to moderate ulcerative colitis. That means we're broke through your walls. So that's another fecal matter transplant thing. What's this one? Oh, this is engraftment. Engraftment means the new cells that you introduce into your body are working properly and starting to rebuild your immune system. Well, they have to get into that fluid-filled highway and start to propagate themselves. And it says engraftment marks the start of your recovery process. White blood cells are the first cells to engraft because they're the ones that need to be there to protect you. And then they followed by the red blood cells and platelets. What's next? Oh, weight loss results in obesity. Differ with clinical microbial factors. There was something about, they didn't seem to feel that it did a whole lot for weight loss. But then I saw other ones that said it did work, but you needed donors from not just one, you needed like 3 or 4 separate donors. There was something about that, about multiple donors. Look at this, fecal effect on stroke outcomes. And it was pretty good, as far as I can remember. Conclusion, systematic review of the preclinical studies showed that manipulating the gut can be a possible therapeutic approach for treatment of stroke and recovery of post-stroke complications. Well, why did the stroke happen in the first place? There was likely a possible relation between actual beginning stroke, because the blood vessel walls were weak and that sort of stuff, because the enzymes weren't there to create the chemistry, to create the strong, tight junctions, basically. All right, this just came out just a few months ago. Stool transplants may boost immunotherapy success in melanoma. Melanoma is a very serious skin condition. It's a killer. In advanced and metastatic melanoma, fecal transplants from healthy donors in advance of the anti-PD-1 immunotherapy led to a 75% clinical benefit rate, defined as complete response, partial or stable disease that lasted 6 months or longer. Results come from a small single-arm study whose primary endpoint was safety, and they found it was very, very safe, and you can see 75% clinical benefit. That's pretty effective. Here's another one. This is fecal microbiota transplantation. This is just from not even a year ago. And this is about type 1 diabetes mellitus, autoimmune-mediated type 1 diabetes. Now, what do they say? Our results suggest that FMT is an effective treatment for autoimmune T1DM. Now, it's effective. So anyway, that's another one. Let's keep on going. All right, I'm almost positive I went over the C. diff. This is a very serious, this absolutely kill you. And it's 90% effective, but survival rate was 94.7% at 1 month and 78.6% at 3 months. There were no serious adverse events related to this FTM. You know, you got to come up and do some research and look into this. And this goes back, 4 or 5 years, 5 years ago. And that's that. What do we got here? Even in preeclampsia, in women going to have birth, they claim that, well, just think of it this way. Patients with preeclampsia showed reduced bacterial diversity with obvious dysbiosis. That means your stomach-gut action is bad. Opportunist pathogens, particularly these different Fucobacterium and Veillonella, were enriched, whereas beneficial bacteria, including Fecalibacterium and Actinomyces, were markedly depleted in the PE group. So it's obviously bacteria. I mean, that's bacteria. There's no other way to look at it. That is bacteria. And that's preeclampsia. All right, this is where it gets into just hearsay almost. This is from the National Library of Medicine, and this goes back to 2020. Fecal myota transplant for rheumatoid arthritis, a case report. It's apparently only one report. It says no previous case of using FMT to treat rheumatoid arthritis has been reported, so nobody's ever done it. We report a case of a patient with refractory rheumatoid arthritis successfully treated with FMT, indicating that FMT may have a good therapeutic effect on RA. Now, they say nobody else has ever had this done. And here's the story behind it. This was a rheumatoid arthritis. Is an autoimmune disease. All right, you cannot protect your bones and so forth, basically. Some anti-rheumatic drugs may fail to treat RA and cause serious adverse reactions. Now here we present a case of a 20-year-old patient, 5-year history of RA, successfully treated with fecal transplant. Successfully, 20 years old, 5 years she had it, so I don't know what to say about that. All right, so now I go and look, and I say, let's see what they got about adverse stuff. And this only goes back less than a year ago. And they're looking for adverse reactions from fecal matter transplants. And they've had almost none, basically. As you go down through the whole thing, there's a chart down here that'll show you the percentages. Look. You see all these zeros down here? Percent with active, well, hold on. I think it was table 2 showed the adverse event. Yeah, table 2. Look at this. Related serious adverse events, 0. 0.083, These are all different studies that were done. Almost no adverse effects. Almost literally none. All below 1%. None have been above 1%. You see this? All these are all zeros. No, not one is over 1%. And these are pooled analysis of all the people that they could find that were doing these studies and the number of people that were involved.
Roger.
Okay, I never waste a chance to do some good chemistry. Watch this. See that? That's catalase. That's what happens. Catalase is in your immune system to kill off any excess oxygen. I just put on hydrogen peroxide. That's H2O2. If I just put water on there, it would just run off. But H2O2 has the extra oxygen, so it has to be driven off, and that's what these enzymes do. Enzymes are proteins, and proteins are just made exactly like this. This is a peptide. Let's just look at this real quickly. All right, this is by the National Human Genome Research Institute, and literally this is exactly what happens. This is what a polypeptide or a peptide chain is made out of, is a string of these little particles. Now this was just updated a week or so ago. Now, this is what's showing you all these different amino acids that are in these chains. And then once it gets to a certain point, I think the 30 to peptide short amino acid, 2 to 50 linked chemicals together, and then they turn into a chain. And then a longer one they call a polypeptide. And the proteins manufactured inside cells are made from one or more polypeptides. So the proteins are the enzymes. The polypeptides are the bits and pieces they put together to make whatever kind of enzyme they want to create at the end. Now let's listen to what they have to say from the National Human Genome Research Project, National Institute of Health, about polypeptides, updated just last week. Okay, this is not too hard to understand. As we know now, bacteria is the source of the enzymes, and some enzymes break down little bits and pieces, and they might be floating around to make these, or these are just squirted out in chains. So they say between 2 and 50 polypeptides. Well, maybe just a bacteria is just going and it squirts out 2, maybe it's going squirts out 50, but then it goes and then it turns into a polypeptide, which is a real long one. So you could have between 2 and 50 of these little links, and then you get into longer ones, and they call them polypeptides. Well, what do? It's just a big long thing here, and all of a sudden it clicks, click, and it says, okay, this is what they call the tRNA, transfer RNA basically. And down here you have the ribosome, which is this whole thing, and this is the messenger RNA. So it comes scooping through here, getting click, 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 click. It's being programmed. It comes out the other side, and it's all set to go to grab onto something with its new program. That's the way it looks like to me. Your messenger RNA comes in, it gets programmed through a series of click this, click that, click this, click that, click, 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 back and forth from this transferring RNA, and it transfers that code. Then that code comes out and goes and does whatever it's supposed to do, good or bad. Break something down, attack an invader, do whatever. And most of these will reattach, I would say, more than likely, or they will go into another cell and program it with its new program. Okay, so we know about the polypeptides and the peptides, little bits and pieces. And this was the adverse, I think this was the adverse effects. Yes, we went through that, and there were very, very few. Here's low income is associated with poor quality dietary intake, and then they probably had a lot of antibiotics and never been refurbished their gut bacteria. So they become a lot more affected by this, and it's very costly and devastating to people's health. Now, this proteomics is a highly sophisticated protease complex designed to carry out selective, efficient process of hydrolysis of client proteins. All of this is proteins and catalysts. All of this stuff is done by these. So we need to look into these a lot deeper. Whoops, the crime of menticide. That's funny, that should come up. Here's another one, autism. Autism symptoms reduced nearly 50% 2 years after fecal matter transplants. That's by the Arizona State University. And as we saw, there doesn't seem to be any